In this video, we will discuss about the ideas of these tumor vaccines. Tumor vaccines can be made of different types, and one of them could be based on these generic tumor antigens, generic tumor-associated antigens that are usually found in most of the cancer patients, uh, and this and the uh, other part of the vaccines, or the other category of the vaccines, is based on mutation profile, or we can say it is customized based on the mutation profile of the individuals. So let us understand what are the ideas behind those kind of cancer vaccines and how they can be made. So the two categories I have already discussed, that is vaccines, or we can say the virus that are genetically modified, uh, that contains this tumor associated antigens, and also and, and other categories, they contains the mutation, they contains the proteins based on their mutational profile of the individuals, so that this kind of viruses that are packaged with this kind of antigens that are packaged within the virus can help in the activations of the immune response. So this this category of the vaccines are then supplemented with cytokines or adjuvants that we have discussed in our previous videos, and they are categorized as peptide vaccines, so that the cytokines will help in the activation of the dendritic cells, and activated dendritic cells will detect those antigens that are packaged within the virus. So that's how this peptide vaccines helps in the activation of the immune system. The other things is that about the other categories is about the autologous tumor tissue or the allogenic PDAC cell lines. That is either these vaccines are being developed from the tumors that are taken from an individual, or from the uh, commercially available PDAC cell lines. So in this case, what will happen is this: the tumor lysates, uh, the tumor lysates of this autologous tumors, and along with the cytokines are injected into the patient, and this is categorized as whole tumor cell vaccines. In this case, one thing needs to be noticed is that tumor lysates that has been injected, either these lysates can be injected along with the cytokines, or the inactivated PDAC cell lines can be injected along with the cytokines. So thereby this approach will help, the cytokines will help in the activation of the dendritic cells, and this tumor lysates will help the dendritic cells to detect the specific antigens that are found on the tumor cells or non-self epitopes. The other categories is about the autologous or allogenic blood. In the second case, we have used tissue, and in the third case, we can use the blood. So in the third case, what will happen is that this when we culture this autologous or allogenic blood, it will help in generation of dendritic cells on its own, and it can be supplemented with the antigen loading, so that the activation so that activated dendritic cells will be increased, and this can be supplemented with this can be injected into orally, or through efficient injection methods. This antigen pulse dendritic cells vaccines can be injected into the patient, and that will also help in the increase of the immune response of the patient against the tumor cells. So these are the in vitro vaccines approaches. In the case of in vivo approaches, what can be done is, after chemotherapy, it has been observed that um, most of the cells are dead after chemotherapy, uh, or irreversible electroporation, such as, or with the help of oncolytic virus, there is an immunogenic cell that has been observed, and that leads to these antigens that are specific to the tumors across the within the host, and this will cause the immune. This will elevate the immune suppression, and at this point of time, some of the immunotherapy such as immune checkpoint inhibitor or adjuvants can be added to the patient so that it will further accelerate the immune response against the tumor cells. And what will happen, so these are the differences between the different cancer vaccines ideas, and rest of the part are similar. So after activation of the immune cells, or activation of the immune cells, or when the dendritic cells detects the antigens or uptake the antigens, these antigen presenting cells will help in the T cell priming, and this T cell priming will leads to the generation of the T cells that can target the tumor cells that have the antigens that has been, those information has been provided by antigen presenting cells to this T cells priming, and after that this T cells can search across the host body and detect those cells that have those cells which have this antigens that has been antigens that has been provided by the antigen presenting cells, and thereby whenever this T cells with this specific antigen information detects any of the cells across different part of the uh, body of the host, it can trigger the immune response and cause the death of the cancerous cells. So these are some of the tumor vaccines ideas that those research are still under process, and this could be the possible solutions for the solid tumors such as pancreatic adenocarcinoma. So I hope you got a basic idea of how these tumor vaccines will function, and how they will help in increasing the immune response of the host and against the tumor cells. If you need to understand more about this tumor vaccines, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.